Hi, everyone. My name is Naveed Tavakol, and I'm a PhD candidate in Dr. Gordana Vunyak-Novakovic's lab at Columbia University. I want to first thank the organizers for having me here today and also letting us talk about our work on an astronaut-on-a-chip, a human multi-organ platform for assessing the extended effects of cosmic radiation. So one of the greatest risks of space travel and intended space travel to the moon and Mars are really the effects of cosmic radiation. And so what this means is that there are a number of different radiation sources that come into contact with either a spacecraft or humans that are in space. And these risks are actually hundreds of times greater than the amounts of radiation that we see on Earth and also in like airplane travel, for example. And so what we're trying to do is better understand these effects in both the context of galactic cosmic rays, which can include a number of different ion sources in space, but also the interaction of these radiation sources on a spacecraft intended to go to Mars. And so the framework that we're really using in some of these studies is really to understand the predicted radiation risks. So what people have already identified over the past maybe 50 years or so on damages to human health that might be a result of any types of radiation. This includes acute radiation toxicities, malignancies of the blood, cardiac arrhythmias, et cetera, that might be in a problem for different human health. And the crowd here is really familiar with the organ-on-a-chip systems and different microphysiological systems that can better help us understand physiology of human health in these contexts of radiation that you might not be able to mimic in other human contexts. Especially animal models, they're kind of the gold standard for radiation research at this point, and people have been using any human system, so whether that's ex vivo cell culture, to better understand how radiation may affect human cells. And what we hope to do here is really be able to take a patient sample or an astronaut sample of their blood, reprogram these cells into iPSCs, and then differentiate these pluripotent stem cells into a variety of different cell types. The goal here is really to envision a patient on a chip where you have one specific organ or multiple different organs interacting together to be able to study different radiation sources, different doses of radiation, and also the timelines, because we're really concerned about long-term space travel and how smaller doses of radiation over a longer period of time may affect human health. And some of these things are a little bit harder to study in vitro systems because you need really those longer time courses. But we're hoping to get there over the next 10 years or so. And so what our work is really focused on is engineering a human organ-on-a-chip system to study the systemic effects of radiation toxicity in this Envision Mars mission. And so we've chosen 3 model systems in this approach: the heart, bone marrow, liver, and then a vasculature barrier, so a compartment to help separate our individual tissue niches from a recirculating compartment. And then study these effects of these different organs to photon radiation and neutron radiation. And I'll go a little bit more in depth into the differences there. We hope to study both acute versus chronic exposure timelines. And so in a traditional setting where you have accidental radiation exposure, this would come in an acute context, but this isn't really a good mimic for what you would see in space, because these are, again, smaller doses over longer periods of time. What we hope to do in future work is really benchmark our engineered tissues and what we're seeing in vitro to any human metrics, so data available from either patients or people who have gone to space and come back, or accidental exposure from different nuclear disasters in our history. And then really to be able to identify radioprotective measures that could be able to mitigate some of the radiation effects that we would see in an intended Mars mission. And so what we did in our study here is really use a source of high LET radiation, which is mimicking neutron radiation. And so these are the effects that you would see when galactic cosmic rays interact with the spacecraft. So not necessarily all the different ion sources that you would see in space, but a portion of them that the actual astronauts would sense. And so we have a facility actually conveniently at Columbia called Nevis Laboratories, where they have these neutron sources and are able to mimic some of the effects of neutron radiation outside of space. And here's just a graph comparing where the GCR spectrum is in space and what you could get more similarly in some of the facilities that people are looking at simulated galactic cosmic radiation. For example, Brookhaven Laboratories in Long Island of New York also has this type of radiation that we're hoping to get to in the next few years. And then the spectrum of high LET sources that we'll see in our Columbia neutron facility. And so my colleagues already talked a little bit earlier in the meeting, Casey and Diogo, on our Organs-on-a-Chip platform. But a basic introduction is that we have a platform that we've developed over the past 5 or 10 years that really is able to take patient samples, whether primary cells or iPSC-derived cells, engineer individual tissues in their own tissue-specific environments, and then integrate them together in a context where you have each individual tissue in its own tissue-specific media, so it's individual niche, and then have a endothelial barrier beneath this that can help separate the media that is for that individual tissue and a common recirculating media. And so the idea here is that you could have each tissue matured separately and then brought together and study some of these systemic interactions in vitro. And so what we look for in our platforms is really to make a platform that's modular, so you can add in any tissue type, mature these tissues over weeks to months, study their connectiveness in the systemic context for a period of 4 weeks, and then try to integrate functional readouts both online and also at the endpoint of our cultures. And so what we're hoping to do is really build complexity to these models. So Casey's paper was published just last month, actually, so you can read a little bit more about the platform itself. But in this work, we're integrating the liver, heart, and bone marrow, which are different organs that were intended for both some of the more sensitive effects of cosmic radiation in the bone marrow and heart, and then liver as an organ that can help metabolize drugs when we put drugs into our platforms. And so just a brief overview of our engineered model of the human bone marrow. We use decellularized bone in a hydrogel system to integrate osteoblasts, mesenchymal stromal cells, endothelial cells, and cord blood-derived hematopoietic stem and progenitor cells. And we can see over a period of 1 week and 2 weeks here that we can maintain the hematopoietic stem and progenitors, and then also over a period of 2 weeks, see myeloid differentiation of these cells into different phenotypes. And so this is just brief characterization of the different stromal populations that are in the tissue niche, as well as the blood cells forming from the engineered human bone marrow. And so just looking at the isolated effects of radiation on the engineered human bone marrow, so without combining it in the systemic context, we see that there's an immediate increase in inflammatory cytokines from the engineered tissue in response to this 1 gray of neutron radiation. We also see the structure of the tissue is affected in response to the higher doses of radiation. And we chose a photon dose and a neutron dose to really compare. Photon doses are more similar to what you would see in cancer therapy, and then the neutron doses are more similar to what you would see in the secondary effects of radiation. And so the neutron doses are about 4 times as potent as the photon doses, which is why we have the differential dose in some of these studies. But we see over a period of 3 weeks that we actually have a reduction of CD45 positive blood cells coming out of the tissue niche over in response to radiation, as well as an increase in median fluorescent intensity of CD45 on those cells, indicating that these cells are either differentiating and becoming larger. And with some of our molecular characterization, we've been able to corroborate some of these findings. So we perform single cell sequencing on our engineered tissues in response to radiation. And we saw that in the photon and neutron doses, there was actually a shift in the population of myeloid cells that was appearing. And we saw a large increase in the population of CD14 positive cells, CD68 positive cells, and a large portion of these cells in this subcluster here is actually populations of monocytes and macrophages that are differentiating towards a M2-like phenotype. So reparative phenotype after 3 weeks of 3 weeks post-radiation. And so the gene ontology terms that are interacting in the system are really extracellular matrix organization and migration of these myeloid cells. And for us, this gives some indication that the system itself is trying to repair itself post-radiation damage. But as we noticed in the previous slide, a lot of the production of the blood cells is actually affected early on with this acute exposure. And so it gives some indication into some of the longer term interests that we have of looking at smaller doses of radiation over longer periods of time, and if this effect is still seen in those cases. When we looked at the effects of radiation on engineered human heart muscle, we noticed, a brief overview over engineered human heart muscle tissue. So over the period of the past 10 years or so, our group has developed a platform where we have a pillar system where engineered iPSC-derived cardiomyocytes and primary cardiac fibroblasts are integrated into a fibrin hydrogel and then electromechanically stimulated over a period of 4 weeks to mature the tissue. And so this is published over the past 5 years or so. And then we have a small adaptation over the past year where we have a smaller tissue format so we can integrate this into higher throughput studies. And so these engineered human heart tissues in response to radiation, we saw actually a decrease in maximum beating frequency and an increase in excitation threshold for these tissues. And so what this means is that the tissue would actually need more stimulation or more electrical stimulation to reach the same threshold as what would be seen in the healthy control tissue. And an interesting thing that we had noticed as well was that the force generation was actually higher in these neutron treated groups. And so this gave some indication to us, and looking also at the molecular characterization, that there could be an increase in early hypertrophic cardiac tissue phenotype. And this is an area that we're actively exploring with our data. And so getting to the point where we actually use the studies of isolated radiation damage and try to integrate this into a systemic context, we created a system where we engineered each of these 4 different tissues with primary cells or iPSC-derived cells, and then integrated them into the systemic context that I described earlier. And so what this means is we have our engineered human bone marrow, as I described before, our engineered human heart tissue, our engineered human liver tissue, which is a combination of hepatocytes and primary fibroblasts in a fibrin hydrogel to mimic some of the metabolic functions of the liver. And then an engineered human vascular barrier where we have endothelial cells and mesenchymal stromal cells to help prevent basically mixing of the media. So we have this individual tissue niche compartment from each individual tissue niche to the circulating media. So we matured these tissues individually over periods of 4 weeks, 4 to 6 weeks, and then integrate them into the systemic context, expose them to either an acute dose of radiation or a protracted dose of radiation. And so this protracted dose is really trying to mimic that smaller dose of radiation over the period of 2 weeks. And then we ended the experiment at that 2-week time point to really characterize what are the effects of what's happening to each of our tissues in response to both an acute dose of radiation and some of the longer term doses of radiation. And so what we saw is we actually saw the same myeloid skewing of the human bone marrow in these systemic contexts. So we noticed an increase in CD11b positive myeloid cells. And what was really interesting is that actually the groups that were treated with the protracted doses of radiation, so smaller doses of radiation, but the same total cumulative dose, actually had a higher proliferation of these CD11b positive myeloid cells. And so it gives some indication and also an increase in myeloid-derived suppressor cells. And so this actually correlates with some of the early findings people are having on increases in myeloid malignancies in response to some of these smaller doses of radiation. but we're doing more molecular characterization to really understand if these cells themselves are inherently changing, or if it's a result of is it a result of the actual dose, or is it a result of the type of dose, so the neutron dose in specific. When we looked at our cardiac muscle phenotype over time, we noticed that early on in the acutely treated multi-organ platform, we saw an increase in cardiac troponin release at that 24-hour mark, but this was actually restored to normal in the later time points, so the 1 to 2 week time points, which makes sense because in the smaller doses of radiation, the cardiac tissues are actually being stimulated with very little radiation over that at the everyday time point, but in the acute dose, they're getting 0.5 gray of neutron radiation at one time. But what we also noticed is that the maximum contraction velocity, like we had seen in the isolated tissue studies, was actually also increased in the acute dose and protracted dose, higher in the protracted dose than you would see in the acute dose, which corroborates some of what we're seeing with the bone marrow, in that the protracted dose of the smaller doses of radiation might actually be having a more detrimental effect to the tissue itself. And then when we looked at the liver, we noticed that the liver actually didn't have any significant, or not many significant differences in albumin and urea secretion, which is what has been seen clinically, and also what people are seeing with animal studies with the same doses of radiation in neutron-specific context. And so taking all of this together, we're really trying to use our organs-on-a-chip systems to really study radiation and the effects of cosmic radiation on human tissues. These patient-specific models of bone marrow, heart, liver, vasculature can answer specific questions, whether that's dosing or the use of radioprotective agents. We've been looking at some radioprotective agents for the hematopoietic system, like GCSF, as well as some other predicted measures that people have used in previous studies for neutron-specific contexts. And then using the crosstalk between multiple organ systems in the systemic context can really enable studies of astronauts on a chip, and taking a specific astronaut's blood, creating iPS cells, and then being able to see that specific astronaut's, the effects of that type of radiation in that specific astronaut over this period of time. And we're really excited for this because it can really inform future studies and what people are really intending to do in space travel as well. And I just want to thank my lab. The entire lab, especially Casey and Diogo, are here at the conference. So if you have any questions, please feel free to reach out to us. And then also NASA's TRISH initiative, which has been extremely supportive in funding this work, but also really helping connect us to the space biologists that are doing these studies in animals, and being able to try to compare what we see and what we see in what others have seen in radiation specific contexts. So thank you so much.
Thanks, Daniel. Awesome talk. Brings me back to being a little boy, and I want to come to Colombia. Are there any questions from the audience? I have maybe some more generic questions. Using iPSC cells, how have you checked how confident you are, how mature these tissues are when you actually put them into your chip?
Yeah, definitely a very great point. So I think we're still developing the right metrics to characterize our tissues. So we've done a lot of work in the cardiac context over the past 5-10 years. And with our other tissues, we're really trying to validate our tissues and benchmark them as close as we can to available human data. And until that point, we're really trying to, in parallel, really study the effects of radiation in the both primary context, but also iPS, and see if there are differences. And if there are differences, is that a lack of maturity of the cells, or also are those cells not performing as well because of whatever artificial systems we're kind of developing? And so I think that's why the benchmarking is so critical in our studies, at least.
Have you already got things set up to get astronaut blood? Was it a wish?
Yeah, so astronaut blood, we haven't specifically, but the goal is really over the next few months and year really is to compare the effects that we're seeing with these neutron radiations to the simulated galactic cosmic rays at the Brookhaven facility I mentioned before. So the idea is at that point, we'll hopefully be at the right, I guess, vantage point for everyone to kind of come together and then either provide astronaut blood or collaborate with the people who are doing some of these other studies in vitro.
So I have a question pertaining to devices and biomaterials. So how do you control for the radiation's impact on the device and the biomaterials and leachables that you might have?
That is a very good question. So we haven't specifically looked at it in this context, but I think for all the materials we're using, they're not actually in our system. They're not actually they don't become radioactive after that exposure. But I think if we're using materials that do become radioactive, we have to do a further step back and see what actually we can mitigate.
I guess
there.
the question is more about what is it doing to the material rather than it becoming radioactive.
Oh, so we haven't looked into that, but I think that's a very good question that we can investigate.
I have a question. Do you also have a possibility to measure for the cardiac for the muscle? Can you measure the strength of the intensity of the contract, the force actually of the contraction?
So I showed some of the functional data, but yeah, we can look at the force as well, and we've looked at that data.
Okay, it would be interesting to use this system also to study microgravity or things, because this has a lot of influences. I was learning recently, also in cartilage and other tissues, in astronauts, they suffer because of that. But of course, it's a different question. So.
Definitely, yeah. So I think for this study, we chose the specific organs we chose for one, to look at the most sensitive or some of the most sensitive organs, and then also in the Trish work, we're really trying to develop radiation countermeasure tools, and so the liver is really critical in this. And I think in future studies we can integrate more organs. It just I think limitation of systems.
Of course.
But, yeah. Thank you.